Okay. Now, it's not going to be about Darwin and her art. This is the real topic for today. Okay, I am nervous because this is a lot of material that I've been working with Katsushi Arisaka, professor in physics and astronomy and electrical engineering for the past, I think we've been working together 6, 7 years at least.
Yeah.
And you've been working on this issue that we're going to get into for over a decade now. And I kind of joined up with him, actually fortuitously, a student that was in his lab, an undergrad, that also took my Psych 110 class and told me I really had to meet Professor Arisaka too, that we probably have some things, interest aligned based on what I was talking about in my learning course. And he was not wrong. That was sad. That struck a collaboration, and I've been more, I like to put it as like, I'm the Watson to his Sherlock Holmes. He's the big brain. He's been really developing this approach. I find it fascinating, and I get it at many levels of conception, but not as deeply as Katsushi does. So I'm glad you and some of your students could be here also. And so just as a backstory, Katsushi developed this elegant mind club, as he calls it. And that's a picture of them at one of those university poster days from back before COVID. And his group presenting. They always have a lot of posters at the event. It's called Elegant Mind Club because his first research was with C. elegans and trying to understand the mind. So kind of cute. But then it developed into working with other species. We jumped from elegans to humans, and then backtracked also to continuing that. So I'll talk about today some of the human work, but also works with fruit flies, fruit fly larvae, slime mold you might be getting into. Yeah, like really comparative, which is really interesting. And I'm going to be focusing on like some of this kind of perspective on encoding space with time and evidence for that in reaction time studies and visual reaction time studies. And that'll be what I'll focus on here, but we have other work going on as well. Yeah. Yes.
Does your lab have a Latin motto?
That's not Latin. It's not Blaisdellius. It's Blaisdell Comparative Cognition Lab. Oh, the below it. Oh, yeah. What is that? Where is that from? I forget. When we came up with that, I forget what that means now. Yeah. Discovery ab animali mente. Okay, yeah. Discover the animal mind. That's how we put it. Okay. Thanks for noticing. Yeah. Everybody's got to have a Latin bio to work. All right, this was the question I think that you started with, right, Katsushi? Why, when we move our eyes, move our head, everything on our 2D perception is moving, it's jumbled on the retina, but our sense is that we have a stable allocentric space. And we could perceive 3 dimensions. We feel depth around us. And we recognize the 3-dimensionality of shapes of objects. And this is just a little video with eye tracker showing somebody as they're driving that they're kind of looking all over the place, and yet they're probably observing, presumably, a stable world like we all do. And one of the early insights that you had, I believe, was what you call metmos. And this is the idea that we actually, in order to do this task, to be able to move around with chaotic input, but have a stable experience of the world around us, you start with the mind first. You start with a model, a mental model, what we call memory. And that's built up through infancy, maybe some genetics, everything, whatever it is that makes the umwelt of your mind, your mental model of like, I'm in a room with a door and these people and a table. This is like my reality right now is this mental model of what's around me. That's the memory. I know that if I forgot to bring my apple or my banana, but I know if I see this and I reach out and I grab it, I know that it's there and I could expect to feel it. Well, how does that work? So that's where we start with memory, and given that the world around me, and let's say I wanted to touch the remote, and I have a part of my mental model, this memory, is that there's a remote located somewhere in space around me. I make a prediction that if I move to my arm to grab the remote, or if I move my eyes to look at something, that I will sense something. I will sense visual input. I will feel tactile input. that either confirms or disconfirms my memory. I mean, this is what we this idea has been around for a long time in psychology. Even the in the 1800s, the German psychophysicists really kind of were coming up to this approach of active inference, predictive coding. The Gestalt kind of approach takes a lot from this kind of early view. So this is just a way of phrasing this idea that, and for humans and other vertebrates, maybe it's higher cortical areas involved with memory and prediction send signals to motor planning areas that involve with motion planning control and to lower sensory areas like an efferent copy that predict what the sensory input will be when the movement is executed. So the sensory input comes in and is compared to the prediction and updates the higher memories via feedback. So the brain is constantly making predictions, and only errors are propagated when reality doesn't match. The things that match our expectations, we just keep moving on and through this MEPMOS loop, continually running at many scales. This is just the gist of the idea that only when I reach for something and I miss something unexpected, I reach for this and it was fluffy instead of solid, then there's something surprising. That's when my attention reorients and I have to do some exploration, something to update my model. See, why was my prediction misaligned with the reality I experienced at the sensory level, at the incoming stream? So to just kind of view this in a diagram format, we start with memory, which leads to prediction of, let's say I think there's a banana in front of me. And so that's my memories or my knowledge state of the world is that there's a banana in front of me. I want to reach and grab it. So I make a motion into the outer world from my internal mental world, and I sense the banana. And the corollary discharge and the sensing all happen at one place in space-time. And so that's what we come to, and that's where Katsushi's first, I think, big insight was that you had to bring in from particle physics the idea of local causality. And so in order for a brain that's a distributed network to keep track of individual things at individual points of space-time, to have a coherent representation, it involves every location within that network obeying the properties of locality and causality. Causal actions only happen at a local point within the system.
What's the alternative to local causality? Is that like quantum entanglement, non-local physical?
That could be an alternative, or it could be just a vague, not a vague theory that doesn't constrain itself to a local causality. I mean, we all believe as scientists, pretty much most of us are of the opinion that, that everything's mechanistic. And we know that everything's happening by causality acting at places where things intersect physically, whether it's a signal, a confirmation of a receptor with a protein, or all that. We know it's local causality, but a lot of psychological theories are vague with respect to this. So if we start from local causality first and build our way up from those first principles, that's kind of the approach we're taking. Do you have anything to add to that, Katsushi? Yeah. Okay. So from this local causality, I want to take a step back and talk about how it's been explained to me how fundamental this was, and comes from, again, from physics. And this idea it originally comes from the physics idea of a light cone, where any point in space-time, causality happens at that location in space-time based on two particles. Like if two particles are going to interact, they have to be at the same place at the same time. And to understand what possible two particles can interact with each other, if the further away two particles are in space, so space would be along the y-axis here. The further away one particle is from another, and if it's constrained by the speed of light, if particles like an electron can travel at the speed of light, then the further away it is in space, the longer it's going to have to travel, the longer the travel time before it can interact. And so that produces this idea of a light cone. That's from physics. showing how particles, the possibility of particles interacting. And if we bring it into the brain in internal space, we know that neurons also have usually a pretty consistent, constant speed. And so this can tell us when we might perceive an object in space with a coincidence detector based on where it is in space and how much time the neural propagation can happen to bring the points to the coincidence detector. That's kind of like the gist of the idea here. So according to Einstein, the only way to measure space or distance is by measuring time. The amount of time it takes, for example, light to travel from one location or point in space to another, that tells you distance because the light is a constant velocity and it's known, and it's also the maximum. So according to Einstein, we measure distance in space with light by measuring with time. And so what Katsushi suggests is also the same thing is happening for our mental experience of space and objects in the world, is that we're transforming spatial information, like on the two-dimensional surface of the retina, into a time code, like a temporal difference between any 2 points in space. So that becomes the fundamental unit of measurement in psychophysics. And that would be true both for, let's say I'm looking at the apple and then I look at the banana, or I look at apples lined up in front of me going off into the distance, or the height of something. I might be moving my eyes to look at them, that amount of time to move my eyes, or the amount of time for brainwaves if I have getting ahead of myself maybe a little bit, but if you have on your visual cortex the retinotopic image there, and you have alpha waves going across your projection in the V1, the primary visual cortex, then those alpha waves are going at a specific rate. So the amount of time it takes for an alpha wave to go from one side to another side of an object kind of gives you now that temporal difference, that phase difference gives you a time. And that time is a derivative of the distance. And it's a compressed form of storage of the space. Yeah, with that. Okay. So that suggests the idea of holographic tomography, or creating a hologram. A hologram is basically a compression of spatial information into temporal information. So if we come back to our space-time diagram that I showed you with the MEPMOS, as we're calling it, loop, memory, prediction, movement, sensation, then imagine that we're sitting there, we have the world around us, that we're our memory of it, our simulation of the world around us. Now imagine something onset suddenly that we didn't predict, we didn't expect was, oh, sorry, wrong direction. Didn't expect was there. Like a flower appears or a light appears. That, no matter where it appears in our visual input, something suddenly comes on, it draws our attention to it. We can sense that, but we don't know semantically what it is yet. We might have an idea, a hypothesis. Or if you're walking along a path and all of a sudden there's a curved stick and that you see in your peripheral vision, you might notice it, and now, was that a snake or a stick? You might have an idea, but you don't know. You have to go and actually move your eyes to it and take a look. And so you have to move, plan to look at that location, send your corollary discharge, and then once your vision gets there, you can see, and it's in your frontal view, you can now see if that is what you thought it might be. Is it a snake or is it a stick? But the detection happens unconsciously. But to consciously understand semantically what something is, what type of object it is, you have to go through this entire process of imagining what it might be, predicting it, moving, and then confirming or disconfirming. So this is an example of this MEPMOS loop at play. Yeah.
Is the motion aspect of it a necessary part of the process? Because you could be paralyzed and have your eyes paralyzed and have images appear before you, and still be able to sort of generate memory guided predictions, and as you gather more information by sampling the stimulus without moving your eyes, you're still accumulating information that can resolve.
That's right.
Prediction.
Motion is necessary, not necessarily overt, it could be covert motion, meaning brain waves.
Oh,
So if you're fixed, if I'm looking at you, I can actually attend to it. Frank just reached for his cup and is drinking now. Like I could actually attend to that, even though it's in my peripheral vision, but and that's the crux of what we're gonna get into with our studies. Yeah, so the retinotopic image now is an image that can be processed internally with brain waves as opposed to externally with actual movement. Great question, because that sets up the direction we're going in with this. Thanks, Frank, for playing part in that. Another thing that's puzzled vision researchers is that we know that when the retinal input comes in, it's still retinotopic in early phases of visual input. After a certain point in both the ventral pathway for like object recognition and the dorsal pathway, the aware pathway for positional encoding in space, retinotopy disappears. And we don't know what code it's been changed into. But the suggestion from this perspective we have is that it would have to be some form of holographic storage code, a time code. So where does space become time? Sometime after these locations in the two pathways of the dorsal and ventral pathway of the visual streams. But then the question is really, how does space become time? Like, how does this transformation from spatial input into time code happen? An insight comes from our knowledge of ring attractors, which had been discovered and are still studied in insects and other invertebrates. Ring attractors are the circular array of like 8 neurons usually that they can operate presumably like an oscillator, and they're like a compass. They can tell you where things are in space. They track spatial information. And you can have multiple ring attractors. You can have a ring attractor for egocentric information, from my perspective on the world, and for allocentric information, like the door to exit is right there. And the difference between those two ring attractors, the phase difference between one ring and the other, now tells you that phase difference is a time difference. That tells you distance. That tells you that encodes space. You can turn that into storing just that phase as a single number, as opposed to maintaining it in retinotopic. And you could think of turning these ring attractors into just these lattices. And so Katsushi came up with this term, holographic ring attractor lattice, or just holographic attractor lattice. These attractor states, which is what ring attractors are. And so we abbreviate it as tau, and intentionally after the 2001 The Space Odyssey movie. Yeah.
So is this theory about what insects do your guys' theory, or is this everybody?
This part
knows
this part is known that they act as rings. What we don't know is about oscillators, I believe. Is that like the current state? We haven't found that in the insect community, neuroscience community, any publications showing that they're acting as an oscillator.
Or speculating?
Or speculation, as far as I know. Yeah.
There's some ring.
Yeah, right. Yeah.
How does an egocentric ring attractor work? So like for an allocentric, the idea is I'm trying to track my head direction. So as I turn my head, making a bump of activity around it.
Right.
But in an egocentric framework, where I am, isn't that always 0, 0? I'm representing everything else relative to me.
That's a good question.
Egocentric means wherever your gaze is shooting is 0, 0.
Is 0, 0.
So my eccentric is moving together with my axis. And then your brain has to convert it to.
No, allocentric would be no matter which way I'm looking, that door is still there. Like it doesn't move in space as I move. Egocentric stuff does, allocentric stuff doesn't.
So if you, let's say you take a camera, the first pixel would be perhaps 0, 0, like that. And so as you're moving the camera around, that position is always gonna stay 0, so. But it's moving around and out.
And so, yeah, you need some other process that holds on to all the allocentric information, like a model that's already built, if it's a camera, and updates where those things are with respect to egocentric. Like if you're mapping between the two, you have to have something that's continually tracking every time you move one way with your egocentric, it subtracts one on the allocentric, because now it's going to be one distance more that way. And that maintains allocentric consistency. That's the gist of the idea, right?
I don't know. Oh, I guess I'll just go a little more.
And in insects, these represent 3 dimensions of space. They're gonna have
Right now, they're one-dimensional. Like they haven't found more than one ring attractor. Yeah. They presumably exist. So by proposing this, they're building a lot of what's known in neuroscience, but there's a lot of things that also haven't been explicitly put in literature yet, but that we predict would be there. We predict that insects should have multiple ring attractors for horizontal space, vertical, and potentially even depth. And humans too, and this is where vertebrates come in, is that in cortex, you can take these kind of HAL structures, this lattice structure, and if you take an X dimension, a Y dimension, and a Z dimension, a depth, and you run planar like alpha waves along those, any object in space will have temporal differences along all those different dimensions along the 3. And if you think of these as synaptic connections, then you could reward any connection when they fire at the same time based on the co-occurrence of these alpha waves as they scan space around us or scan the visual input in a retinotopic map. And so what does this get you? If we propose this theoretical construct is actually what happens in the brain, well, now you can have your XYZ axis, your 3 dimensions of space as a set of connected synapses. And so this would be your memory of the banana or your view of the banana directly in front of you. If the banana is shifted a little bit in your vision, then that's just a simple linear translation along those 3 attractors, the 3 HALs. Likewise, if you're looking at one thing, you're looking at another thing, or moving through space, again, if you represent the apple as one location in your retinotopic vision and the bananas in another location, they have a relationship to each other. And if you look back and forth, you could just translate them with respect to each other on these attractor lattices. It's simple linear translation. And so the idea is, how would you test this without actually opening up the brain and looking for these things? Well, the first immediate thing is that if this is true, that that is stored as a time code and scanning involves time, then the more, if you're looking at something in dead center and something else comes on in your periphery, in order to figure out what it is, the further away it is in your periphery, the longer it's going to take your eye to move to it, or the longer it's going to take for a brainwave to move along your retinal scan and then scan it and determine what it is. So for semantic processing, there should be, to make a choice about, is this the apple or is this the banana or anything like that, it's going to take longer the further in the periphery it is. Whereas if you just are reacting to tell me when something comes on, if it's in the center or on the side, that should pretty much be flat, because that doesn't, you're not making a decision about what it is. That doesn't require all of those. Let me go back. That can go as a simple reflex. Whereas if you want to process the meaning of something by turning it spatially into a time code, then by following this neural holographic tomography and encoding it as HALs, am I going the right way? Going the wrong way. That takes time. So that leads us to the prediction that simple reaction time, just press a button if something comes on in your vision anywhere. You should be pretty flat and stable and pretty fast. But to make a decision about what something is, it's going to take longer. because you have to do one more movement through the down here, complete the Metmos loop. And the further it is from your center of view, the longer it's going to take to process that. Yeah. Yep. And there might be variations in different, like alpha in one location might be at 8 and another might be at 9. As long as it's consistent at each step of the way where this would be happening, that should work fine. Yeah. Yeah, so alpha phase precession would be a lot like theta phase precession in the hippocampus. And so to have a sense of space and everything in it to using these kind of approach, you really need 7 degrees of freedom. 7 of these linear scales. There's 3D for space, 1D for scale invariance. So we'll see like scaling up or down, and 3D for rotation, like the let's see, roll, pitch, and yaw of like the human face, the human head, or any object rotating in space. And the idea is that because we're comparing our visual input to something about in memory, we remember something in a specific, like right in front of you, and a specific size. And that's like my memory of this water bottle, which I've been using for a couple of years now. I have a really good memory of it. I can, if I close my eyes and picture it, usually I'm picturing it kind of like right in front of me, and of a certain size. I'm not picturing it all the way across the room or off to the side, where if I'm looking different, my imagination would bring it up right in front of me. So if you encode if your engrams stored as HALs are of an idealized version of something, then anytime you see something in your periphery, oh, is that my water bottle? You have to go and look, or you can process it with alpha waves or something. It takes time to bring it in your mind's eye to the center to then compare it to your stored memory. And that's kind of the gist of what's going on. You're comparing to memory what you're seeing, what your senses are telling you. And so now I'm going to transition to showing you 4 different studies where they're all variations of the same kind of idea. They're all going to be testing ideas about the invariance in space, invariance of scale, and invariance of rotation, a 3D rotation, with human reaction times experiments. And these are just a subset of many different studies that have been going on in the Elegant Mind Club lab for the past many years. One of the early apparatuses that we built is, we call it a zip dome. It's this geodesic dome kind of in front of the participant, seated in front of it with a little chin rest. And it's called the zip dome because it's all these zip ties to tie it together. And along each one of these runners is a set of LED lights. And so what you can do is you can have the participant seated there with the chin rest in their hand on a little button press. And you could do 2 types of studies. You can have them do a simple reaction time task where you tell them, as soon as you see something turn on along these lights, just press a button. So simple reaction time should be pretty fast, and it shouldn't depend on any eccentricity. They're asked to look at the gaze at the center. There's a little fixation point. And so it shouldn't matter whether it's off to the periphery or close to the center. It should be really simple and fast and roughly equivalent reaction time. Alternatively, if they're given a choice reaction time task, then they're either going to see 1, 2, or 3 lights come on. And there are 3 lights right next to each other. Is that right? How it worked? It's been a while since I've seen this one. So 1, 2, or 3 little blue lights here, or 1, 2, or 3 blue lights here. So anywhere in that location, we can look along the X or the Y dimension and see whether the further from the center of view it is, the longer it takes them to make a choice of which one that they see. And so simple reaction time should be flat, a function of reaction time by eccentricity, and we should get a V-shaped pattern for the choice task. A similar study, go ahead.
Doesn't the sort of density of photoreceptors of the fovea factor into something that's more peripheral, your detection of a feature of it, like what color it is,
Yep.
As opposed to whether or not something flash, which might be.
Right. So that is an alternative account for this particular type of study. That's where the other studies looking at rotation and scale invariance will be important because they don't suffer from that alternative, as far as I understand. But yeah, it's good. That's a good criticism of this, that it's opened up to that being an alternative explanation.
で、the two of you, we see this pattern in polar coordinate system. We should have a very steep curve in time. This, but what we found is she's going to completely linear. So, and that makes sense. Our visual perception is linear. Even though a pattern we receive on visual cortex has dense information at the center, we somehow convert to the linear.
So that pattern on visual cortex is because that's the pattern that it receives from retinal input, for the reason that Jesse's bringing up, right?
But we don't see a pattern on retina. We have to somehow convert logarithmic scale or dense information at the center to the flat space by the time it comes to the frontal cortex.
Okay.
And we'll see another approach that we use is with the computer screens. In fact, now we've moved to VR headsets for this kind of research, but we could do also the same with computer screens with a chin rest and a keyboard. And you could either do a simple reaction time if you see something appear, and those things could be faces or Gabor patterns or a letter. No matter where it is in the periphery, center or periphery, if they're just told press a button when something appears on the screen, we'll see that, and we predict and we'll see that they're a very flat response, depending, and it doesn't depend on eccentricity. Whereas if they have to make a choice of like if they've been trained on 2 or 3 faces, or asking about a Gabor pattern, is it a left or a right tilting Gabor pattern? Press one key if it's left and the other key if it's right. Any kind of choice like that, we predict a V-shaped pattern as a function of eccentricity, which is what we're showing here. Relatively flat and fast for simple reaction time. That's the lower set of dots in each of these graphs. And the BBB, horizontal and vertical, that's that zip dome. So either along the horizontal or along the vertical axis, seeing 1, 2, or 3 blue lights. The further it is in eccentricity, the longer it takes, but it's a linear scale, a linear function. And that's true for faces and letters and Gabor patterns. Yeah.
I can sort of capitalize on Jesse's point. I think you might be measuring just the time to make an eye movement. Because you have an answer for this question. Okay.
Yeah, that you cued a memory. I should have said, I forgot. For some trials, participants were allowed to move their eyes. For others, they were told stay fixated on the fixation point and press the button. Either press the button, you're allowed to move your eyes, and we actually measure eye. We have, yeah, and that's the covert or overt. I forgot to mention. Thanks for queuing me up. I forgot to mention that. They look the same, whether they're just fixating, maintaining fixation on the center point, or even moving their eyes. Yeah, we get the same result. I forgot to include that in here, but yeah.
The speed of eye motion is identical to the brainwave traveling inside of covert attention. That's why covert attention, but eye motion at the same time.
And it was constant
That's why
velocity.
No matter how you move eyes,
We also did the same for depth, and these are 2 of the different apparatuses. Some people, this was some of this during COVID, so people were at home and they constructed their own apparatus. They hooked up an Arduino to sets of LED strips. They would sit on the floor and they could look down, and with their Python script, they could make it so that the 2 lights would appear at different distances, at different depths, and they were told to fixate, have a fixation point like shown here. So you can have things appear near the fixation point, closer or further away. This was an apparatus doing basically the same thing in the laboratory. And again, you could either do a simple reaction time, just press a button as soon as something appears, or you could do choice. And that was single versus two LEDs flashing. And for the simple one, just respond if you see something, and the fixation point's right here, just around 1. There was a relatively flat, a little shallow, a little bit going up with distance, closer to or further away from the subject, but very, very flat. Whereas with a choice reaction time, it took longer and a much steeper curve and linear function of space. And no difference between overt or covert. So they either had to stay fixated on the fixation point, or they can move their eye, look up and down to where the stimulus appeared. We did the same thing with rotation in 3 dimensions, pitch, like up and down, yes, no, yaw, or no. So yes would be pitch, no would be yaw, and then roll be, oh, that would be isn't it in Indian people, sometimes they say yes, like, I've seen that actually, it's interesting. So all the ways we communicate, we use pitch, yaw, and roll. It's kind of interesting. But we also did this on a computer screen. They first learn to learn, like, let's say these 3 faces. And those are the actual faces that we used. And they learn to press 1, 2, or 3, one a second or a third button corresponding to which face. And so once they learned really well which button to subscribe to which face, then you could present the faces at different amounts of pitch, yaw, or roll. And again, the more the transformation needed to bring the presented face to what you remembered seeing, the longer that takes, the longer, or the more that rotation needed, the longer that should take for them to make the choice. So again, looking at this linear translation, here's the face at the 0 degrees of rotation in either of 3 of those directions. And then as you see which one's which. So pitch had the steepest up there, up and down had the steepest curves, and then yaw had the second steepest, and then roll was the least steep. But still, they all show the same effect.
But it looks predicted to be different for these different transformations.
Not really, except there are more features visible in the roll case. So maybe having more of the features there allows you to recognize it faster. Whereas with yaw or pitch, you're transforming the features the way you normally would see them, like 2 eyes, nose, and a mouth, are being like really transformed more. So you have to do more within stimulus transformations as well to track them. That'd be my best explanation for that. You think so? I don't know.
Rotation.
Okay. Oh, so the translation's happening in log polar, not in Cartesian space.
The mean scale is linear, is moving one direction, and also rotation is the other direction.
Oh,
So it's a
okay.
conversion to look
Okay,
for
okay.
explain why look is easy.
Okay, we'll have to, I don't know if we have that in our paper because we're currently writing these up for publication. We should mention that, explain that in the paper also. Why am I looking at? Mingda's over there. So Mingda, you're making notes? Thanks. Great. I will add that also. I forgot about that. Yeah.
You have to train.
Depends.
Trials of just training before we even like start taking the data. Just so like because you can see like actually all 3 faces right there. When you have them make the choices, they have to like immediately know which button to press for which face. So it takes a while, so it while. But yeah.
That's not a lot though. Like for human psychophysics studies, 50 trials isn't too bad.
Yeah.
Jesse.
Yeah, so you cite Shepard and Metzler's seminal study to mental rotation of these block-like 2D drawings, blocks that you have to compare, and the angle of rotation and depth plane or the picture plane predicted perfectly linear, but the reaction times in that. And then all of the work that Stephen Kosslyn and his group later went on to do with imagery, with looking at scanning of attention across space or zooming in or out. We get these really nice relationships between reaction time and how long it takes to either do the operation in mental rotation or the zooming or scanning of attention. All of that is framed in terms of this. You create sort of a model of the stimulus, and then you apply some transformation to it that takes time and proportional to how much in proportion to how much you transform it.
Right.
How is this sort of account different? Are you saying, is it just sort of a mechanism of how you accomplish
It's
it?
the mechanism. Yeah, so this is, again, you're right. There's consistent evidence supporting this mechanism, but it's never been framed in terms of like exactly this holographic storage of spatial information. Right. And I guess that's the big idea here is it's the only way we can think of that the brain can do these processes with the speed they're doing and in the form that they're doing, is that by turning spatial retinotopy and spatial code into the time code, and then that allows for these linear translations, linear effects. Mm-hmm. I don't remember. It's been a while since we looked at that literature. I don't remember if they talked about how the brain's doing it. Does Kosslyn talk about how the brain's doing it, or just that the brain's doing it?
Broad theories that are sort of reusing this perception during create some modeling entity that you're trying to maybe rotate or in on, and then you're applying some algorithm to rescale it or shift your spatial attention. But it's really about the spatial attention or mental simulation. And he looks at the brain. I mean, he's done imaging studies, and you see visual cortex activity when people are doing this imagery, and spatial attention mechanisms in the parietal lobe, but it's not mechanistic beyond that.
It's not mechanistic to the point of building up like, oh, the transformation happens and here's like a lattice structure, and this is how synaptic networks connections form such that you can store an ideal code for something as an engram. Like it's
Definitely not like that.
yeah. Yeah.
Futures.
Well, in that sense, he's right. Yeah. Exactly. Yeah. Right. We try. The tough thing is getting them to hold still so that we know when something's presented in one place versus another, that they're seeing it, and that there's something called eccentricity, rather than if they're like, because they're looking around like, and that's the thing. We try to do it by having them peck at a stimulus, and in the midst of pecking, it causes something to turn on. And we actually got some sort of interesting data. One of their pigeons we found out had real, like, maybe he's partly blind in one eye or something, because it was all one-sided. And the other two pigeons, we started to see something that looked like interesting, because pigeons have two phobia in each eye. They have one that's myopic. It's for pecking seeds off the ground, and that's where there's a little bit of overlap between the two visual fields. And then there's another one that's more for distance vision, and basically is looking like that. And what we actually found is, compared to the 0 point, we saw two V's. That's not really strong yet, but we need to do more data, more research to support it. But it looked like each phobia, where it was looking, was where the center of the V was. But we need to go back and I don't know if we'll have time to revisit that, but at some point I'd like to revisit that. But animals are challenging to work with this kind of thing. Monkeys would be easier because you can head fix maybe a head fixed mouse or a rat that has some way of doing a response. But yeah, pigeons were tough. The final one I'll present is the scaling. And so participants came in again with the computer task, and they learned either about faces, or they were shown Chinese characters, and these were non-Chinese speakers or readers, or English characters. And then after learning them at a standard, we could then present them at various sizes, scales up or down. and to see how long they recognize, oh, that's a B versus a P, or what were the three letters, for example?
E, B, and
E, B, and P. So you had like three keys, one for E, one for B, one for P. There was also three Chinese characters. Was that how it worked?
yes. I don't remember those though.
And then yeah, right. And then the three faces. And again, wait, yeah. So again, this is the 8 degrees was what they memorized during training. And then when we varied it smaller or larger, then we see in both directions reaction time gets longer, the more they'd have to scale it up or down to match the remembered image. Oh, except for English, because English characters were pretty flat. And it seems like they're scale invariant. Your recognition of the letter B doesn't matter if it's really big or really small font in general. You see most of the lines are flat, and it's only when you get to the extremes, extremely big or small, that you get departures from a flat function. That's basically what I decided to present for today. So I'm happy to have more discussion. Thank you. How much time do we have? Like, where are we at in time?
You go. 10 minutes or so.
Okay.
Like
Hmm.
people with aphantasia, or people with like, I don't know, Meniere's disease, but
I don't think we did.
We did a couple of exclusions, mostly for like visual acuity, but we didn't particularly ask for aphantasia or stuff like
I have
that.
an idea about that, specifically aphantasia.
I personally have ADHD, and we took the choice reaction time and the zip dome, and we tended to have a flatter line.
Same as me. So we.
Really?
Almost-
But did you systematically like ask people?
No,
Or
we
just kind of a handful? Okay.
Yeah, at least for us, yeah.
Yeah.
It was flatter.
Much flatter.
Yeah.
We also knew what you were expecting, which is kind of we'll go against that and
After the data taking, we didn't expect anything. 40 participants outside.
Yeah, yeah. But like unbiased participants, it's just like if they couldn't see very well, sometimes even or they just didn't take data. We didn't just take tests.
Aphantasia is an interesting one. That's the inability to experience mental imagery. Right? I think people with aphantasia predict basically this is how the brain works. They still are doing this. They just somehow don't have their own access to them doing this. Like they still remember things. And then when they see something, they recognize it if it was something they remembered. So it would still presumably go through the same process. I just think somehow they're not able to engage the imagination part for some reason. Right.
That's almost like kind of like blind
Blind
sight.
sight.
Yeah. It'd be like that. Exactly.
Yeah.
Good analogy.
Yeah, I mean, that's fine. Objective experience, they don't describe it in that
Right.
way as being visual or picture like.
Mhm.
But the brain.
Past
doesn't have-
performance. The brain didn't suggest that
It's
they,
not- They actually have the experience,
yeah,
just that they can't report it.
yeah. Which
Kinda like blind
is like,
sight, okay. Yeah.
that it's.
Yeah, I think what we're really interested though is in people in publishing this idea about like the neuroholographic tomography and this kind of synaptic network, and to really spur neuroscientists who, I don't know who would be the kind of people that would have the tools to look at this kind of for evidence of this kind of thing, but that would be interesting to us, like attractor lattices in cortex. I think the best that we've seen so far, people looking at coming towards this approach, there's like Jeff Hawkins talks about cortex as being processors, almost like little mini insect brains. I think he has a book, 1,000 Insect Brains, is like or his view is talking about the human cortex. That might be along the lines of of where we're thinking. Also because of the connection between the cortical thalamic cortical loops, where thalamus can assign phase, and that could do translation phase assignment would be the way that it would drive cortex to do these kinds of linear translations. Yeah.
Going back to the alpha question, but would you expect any like individual differences in like alpha frequency to maybe relate to this? I know it's like also differences in like individual peak frequency related to processing time, so that really hard to disentangle, but that could be a way to.
We actually tried testing this. Our EG devices unfortunately are not good enough. So our data was too noisy. But we did try testing to see alpha with peak frequencies during like eyes closed, and then trying to correlate that to reaction times to a couple of our different experiments, choice, simple reaction time, and then also eccentricity. We just weren't able to get good enough data to find anything.
I think the variation, 10
Yeah.
hertz plus minus 0.5 hertz.
Within an individual.
Yeah. It's like
And
10.
as long as it's consistent within an individual, individual differences, I would imagine wouldn't.
Well, no, 10 or 20 different subjects to measure the alpha frequency of peaks. Yeah. Maybe I think the variation is 5%.
From my memory, it was a little bit larger. But
Larger?
I'd have to go back to the data.
So that's a good point. Of course, we should see a correlation of the slope and frequency. We can find because variation of alpha frequency was imagine.
Yeah.
Presenting the stimuli, if you're measuring alpha in real time, and then you present the stimuli either at alpha peak or alpha trough or in between, you might be able to find effects that are much
That's a good point.
We also tried to phase correlate instantaneous alpha phase with the reaction times, but getting instantaneous alpha phase is a very noisy trial. So like even averaging over like hundreds of trials, it's like hard to get that.
Okay. Thanks, everybody. Okay, so you said the